Good morning, everyone. We'll give folks a second to find a seat. I'm Chris Rebelo. I'm the Senior Director of Communications here at the Buck Institute. It is my extreme pleasure to welcome all of you to what is our 9th event of our Live Better Longer seminar series. Today, we're going to learn about efforts to boost brain resilience to restore memory. So, a couple quick housekeeping notes. If you haven't put your phone on silent, please do. And then also, I like to keep the fire marshal happy. I'm pointing out the emergency exits at the back of the room. So, question, how many of you are new to the Buck? Always exciting to have new people. Instead of me talking about our exciting science, we're going to show you a short video that kind of gives you an overview of what happens here at the Buck.
The Buck Institute is interested in understanding aging, not only to make us live longer, but to actually alleviate and suppress the development of diseases.
Chronic disease is the great health challenge of our time. Aging is the main driver of chronic disease. To eliminate chronic disease, we must harness the biology of aging so we can live healthy longer. The Buck Institute is the world's first research organization dedicated solely to the biology of aging. We work with the building blocks of biology to understand how we age.
The Buck is concentrating on really trying to go to the root of the problem, so understanding that we can sort of intervene really early.
The Buck Institute is probably the best place in the world to be working on aging because there's so many different aspects of aging that are being studied. It's key because no one lab can do it all.
Our scientific explorers are brilliant, curious, and committed.
The idea that you could actually develop drugs that slow down the aging process was really something that 20 or 30 years ago people thought this cannot be done. Not only can it be done, but we will do it.
The pieces are in place. And we are in a unique position to put them together. Our world is defined by paradigm shifts, created by scientific discoveries. The biology of aging is the next frontier.
We're working toward a world where healthcare is better. It's a world where if we can eradicate the aging component to disease, we're all going to live healthier and longer.
It's just an incredible vision that really will, surpass anything else that we've ever seen before.
We are inviting you to a future of aging without illness. It's happening today, and it's happening here. Join us.
So before I introduce today's speaker, I want to thank our sponsors. They are listed in your program. I also want to make sure all of you have note cards, so in case you want to ask a question, just write your question down and a volunteer will pick it up and get it to me. And you can ask a question at any time during the event. Time to make a special welcome as I look at the camera and welcome everybody who is on Zoom. You too can participate. Just put your question in the Q&A section on Zoom and somebody will get those questions to me as well. So now it is my pleasure, whoops, I went the wrong way with this thing, of course. Whoop. There we go. Woo-hoo. Okay, now it is my pleasure to introduce Dr. Tracy. So, her lab is taking an out-of-the-box approach to Alzheimer's and dementia. They are working on a workaround around brain pathology. Very exciting. We're going to be talking about that in a minute. Tara received her PhD in neuroscience from the University of California, Berkeley. You can read about her background in your program. I just want you to know that she's received a lot of awards, both from organizations that are focused on neuroscience and those that are focused on aging. We are fortunate to have her here on our faculty. One final note, Tara has a PhD. She's not an MD. So this is not a clinic. So if you have questions, please make them general in nature, and do not take anything that is said here about lifestyle changes or anything. Don't do anything without talking to your doctor. That's kind of our disclaimer as we get into these things. Okay, Tara, come on up. Let's get started.
Great.
Okay, so Tara works on diseases called tauopathies. Start us off, Tara.
Yes.
Talk about tau and what it is and what it does.
Thank you, Chris. So I would guess everybody in the room knows Alzheimer's disease. This is the most common type of tauopathy. However, there are a number of other diseases that are similar to Alzheimer's disease, but different. And they're all classified as tauopathies because they are characterized by the accumulation of this tau protein in the brain. And there's a picture on this slide showing this pathological tau protein accumulation in the brain that's found in all of these diseases.
So one of the diseases that's listed here is CTE, see if I can pronounce it, chronic traumatic encephalopathy. Not bad. So this is the stuff that football players get, right? And boxers. So talk about that.
That's right. So this is a type of neurodegenerative disease that occurs after brain injury. And we hear a lot in the news about football players who end up with early onset dementia. And it's often because of CTE. And when they pass away, what neuropathologists find in their brain is a whole lot of this tau protein that accumulates. And so it's possible, because tau is a common pathology across these diseases, it's possible that the dementia that's happening is because of this tau protein.
So we weren't able to, I have a video that is all about concussions and about tau. We weren't able to play it today, but I'm going to include a link to that video in an email that you will get following this, either later today or tomorrow, that will have a link to today's session and some other background materials. So be aware of that. So I think most people are aware of amyloid plaques, which are also involved in Alzheimer's. What's the difference between tau and amyloid?
So these are two different types of proteins. However, amyloid beta plaques are specific to Alzheimer's disease. They do not occur in the other tauopathies. So the only one on this list that has amyloid beta plaques is Alzheimer's disease. The others only have tau pathology, which is often called tangles. So if you see the word tangle in the brain, that's a tau pathology. And so these different diseases, one of the biggest challenges we have is determining which disease people have. And so because the best way we currently have is determining that after somebody has passed, and then a neuropathologist looks in their brain. And so one of the things that people are trying to do now is come up with ways of, while people are still living, to determine which type of disease they have.
So How does somebody get these tangles?
Do
they know, or is that still the big question?
Well, many of us have been studying this for many years. We think that tau is a normal protein in our brains. We all have it. And it's expressed in all our neurons. But something, and it could be any number of factors, causes it to become toxic in these age-related neurodegenerative diseases. It changes, and it starts to aggregate, and those small aggregates can start causing dysfunction in neurons. And that's what we think is a mechanism that's causing memory loss and cognitive impairments.
So, your work focuses a lot on synapses. What are synapses and why are they so important?
Synapses are my favorite thing.
Now
I love synapses.
Now you know what to get her for Christmas. Okay, go ahead.
I have many of them, so it's okay. I don't need any more. They're so fascinating. We have trillions of them in our brains. They are the connections between our neurons that are required for cognition and basically everything we do. They have a chemical signal that tells neurons when to become active. And this is what we call transmission. So it's transmitting information from one neuron to the next. And this is what gives our brain the computing power that it has. And the other thing about synapses that my lab really hones in on is that their activity is responsible for encoding memories in the brain, is what we think is happening. And so that's where I started my work in neuroscience. My lab is ongoing to looking at synapses and Alzheimer's disease.
So, synapse dysfunction occurs at the early stages. So talk about that, kind of.
Yes. So Alzheimer's disease and other tauopathies are progressive, which means they start out with kind of mild cognitive impairment, and then it gets worse and worse over time. And we think that the underlying things that are happening in the brain are also progressive, meaning that the earliest stages, we think the little connections between these neurons at the synapses are starting to dysfunction. They're not working properly. And then they start to become lost. So you start to lose synapses. And then at the end stage of the diseases, in the late stage, is when the neurons themselves, the cells in the brain, start to die. And so I think it's really important to think about how these diseases are progressive when we think about how are we going to treat them. Because it is difficult to treat, at the more moderate to severe stages of dementia. But we think synapse function could be a good target.
Your goal, and I love
Yes.
this image. It really just says what you want to do here.
Yes. So I just want to, maybe explain why do I have a forest there. Well, I always think of neurons are so beautiful. They have these long processes called dendrites that kind of make it look like a tree. There's like a little cell with a tree coming out of it, and then the synapses are like the leaves on the tree. And so here in this picture, the model that we have is that this toxic tau builds up the brain in tauopathy, and it causes a synapse dysfunction. And so you can see in this picture of the forest, the leaves on the trees are not so happy. And we think that this is causing memory decline or memory deficits in the disease. And then can we come up with a strategy to repair these synapses and make the forest healthy again in order to restore memory? And the really cool thing about this approach is that if we can repair the synapses, this could promote resilience to the kind of toxicity that happens in the brain in these diseases. And the toxicity I'm talking about is the tau and the amyloid beta protein. Can we make the brain resilient to that?
So that's the end run, right, that you're
Yes.
trying to do. Got it.
Yes.
So I think kind of begs the question, why should we make a therapeutic? Why not? But I'm mostly interested in that first bullet point where you say you want to develop new therapies that are not disease modifying, that there's a potential for broader impact. So if you're successful, assuming you're going to be successful, what's the best case scenario? What are you hoping that you might be able to accomplish?
Yeah. So if we can have a synaptic targeted therapeutic, potentially it could be applicable to maybe not even just Alzheimer's disease, but other neurodegenerative diseases that involve synapse dysfunction. Potentially even just normal memory loss with aging, because there is a component of memory impairment as we age. That could maybe even be targeted for intervention as well. The reason I have this bullet point here is because many of the approaches taken so far with Alzheimer's disease have been disease modifying, meaning they're reducing amyloid beta levels as a major target, or reducing tau levels. And so our approach is different from that. As I previously said.
So this is a good question we got from Zoom. Why do you believe the tau protein is a cause and not a result of the body fighting disease?
Yes. So there are a number of lines of evidence. One of the most important is that there is a very small number of people who have dementia, I would say, that is caused by mutations on the tau protein. So these are familial frontotemporal dementia cases. There are tens of mutations on tau actually that can cause familial dementia. Most people don't have this, but if you have a mutation on tau that's associated with this, you will get early onset dementia. So tau by itself is sufficient to drive dementia.
So let's talk about the inner workings of actually what happens at the synapse. How do we form memories?
Yes. So this is an image of a synapse. There's two different neurons that are coming together here. The neuron in the top is sending the signal via those little yellow dots, and the neuron on the bottom is receiving the signal with these little antennae that are called receptors. And when there are certain patterns of neuronal activity in the brain during learning, so you're learning about synapses right now, so hopefully this is happening as we speak. There's patterns of neuronal activity happening in your brain that are causing a change at that synapse. And the change is the trafficking of these receptors into the synapse to boost the signal. It's like putting more antenna there so you get more of the signal coming through. That increases the activity of that neuron and strengthens that synapse. And we think that this is the basis for memory formation in the brain. And so
Moving on.
Yeah,
what does that toxic tau stuff do?
Right. So work that I've done and also others has suggests that toxic tau that's found in disease inhibits this process. So when you have toxic tau at the synapse, it blocks the trafficking of these receptors, and therefore the synapse is not strengthened, and you don't get that formation of a new memory.
So the big questions.
Yeah, so our big questions are, can we repair this process? Can we reintroduce we call it plasticity, this activity-dependent strengthening of the synapses in order to improve people's ability to form new memories? And also, whenever we think about a potential therapeutic, it's important to think, how do we identify people who would best respond to that therapeutic? So that's another goal we have.
So we are now at the hero of this story, a protein called Kebra. Talk about Kebra.
Thank you, Chris. So Kebra is given its name because it's a protein that's found in the kidney and in the brain. What it's doing in the kidney it could be interesting, but I'm horribly biased, so I have been so interested in really focusing on its role in the brain, and there is a number of reasons for that. It's a synaptic protein, and it was first gained interest in the field of neuroscience from a paper that showed that genetic variation, so a certain nucleotide in the gene that encodes Kebra that people have, is associated with human memory performance. And so people who had a specific nucleotide in that gene tended to have better memory. Later, it was found that that variant was also associated with risk for Alzheimer's disease and for cognitive decline and aging in general. So there's a link between Kebra and memory, and there's a link between the Kebra and the risk for Alzheimer's disease and aging. We also have mice that are deficient in Kebra, and we know that they have memory impairments and they have impaired plasticity at their synapses, which is the mechanism I told you before. So we think that Kebra is critically important for that process of strengthening the synapses to form new memories. It's also kind of a master regulator at the synapse. It binds to a bunch of different things. So that means that it could have big signaling power.
So, Kebra deficiency in the brain is not a good thing. It underlies memory loss. Talk about that, and also in the context, I think well, maybe you're not aware, but there's a sizable percentage, I think it's 15 to 17% of people, when they do post-mortem autopsies, these folks did not have dementia, but when they look at their brain has like amyloid plaques and tau. They should have Alzheimer's, but they don't. So are we capable of being genetically inclined to have more Kebra? Is that protective, or what's going on here?
Yeah, well, this is something we're very interested in studying. We're wondering, why are people with a certain polymorphism in the Kebra gene, why are they at less risk for Alzheimer's disease? And one of our hypotheses is that they might have more Kebra at their synapses to protect them against the cognitive decline. But yeah, previously what we've shown so far is that in Alzheimer's disease, people who have dementia have lower Kebra levels in the brain. And we think that the loss of the Kebra from the synapses is driving those memory changes. Yeah.
Great. So, things that you're working on. So you would like to see Kebra as a biomarker?
Yes. So as I kind of alluded to this earlier, we would ideally like to learn, which people might have lower Kebra levels in their brain and might be more susceptible to memory loss associated with synapse dysfunction. And so we're trying to develop ways of detecting Kebra in living people, and changes, potentially even changes in Kebra with age. And then the second thing is to better understand that polymorphism I mentioned, and to kind of use that information to determine who's more susceptible to the loss of Kebra. And then finally, with these two pieces of information in hand, then we can determine who would best benefit from having a KIBRA-based therapeutic that we're developing.
So one of the questions we got, and I'm going to ask you to talk about it more in depth in a bit, but folks want to know about the new drugs for Alzheimer's,
yes.
And what your take is on them.
Yes.
I know when you and I have talked, we've talked about having therapeutics need to be personalized. So talk some about the existing drugs that are out there and kind of what's the future for treating Alzheimer's, both from like the amyloid plaque, the tau, the resilience.
Yeah, these are all very big and important questions. The last few years, probably everybody has kind of heard about these things. There are new drugs coming out that target amyloid beta that were FDA approved. Two of them are the latest one, the more recent ones are lecanemab and donanemab. These are showing benefits in clinical trials, which is encouraging. They are slowing the rate of cognitive decline in people with Alzheimer's disease. And so that's a good thing overall. There are also trials trying to reduce tau levels in the brain. And so far, they've shown, especially a particular one called the tau ASOs, they've shown good safety characteristics, meaning they've been pretty safe for people so far, and they are in the results from the smaller trial that have been published so far are trending on showing slowing of cognitive decline as well. So these are all good things, or like steps in the right direction that are encouraging. I think, Chris, what you asked me, I mean, the thing that I think that's still remaining that we're trying to address is, okay, it's great if you slow cognitive decline, but can you actually restore cognition once it's been lost? And this is going to potentially be difficult, but I think it's really important that we start to address it, because I do see promise with the drugs that are slowing decline. But I think we can do even better. And in terms of personalized medicine, all of these drugs, I think, and actually, I think now in order to, like, if you wanted to be treated with one of the amyloid beta drugs, I think, and of course talk to your doctor, but I think you have to be tested for amyloid beta in your brain first. They want to make sure that you have the biomarker that suggests that you have Alzheimer's disease pathology. So anytime you get treated with one of these drugs, you have to have a biomarker to determine whether this is the right treatment for you. We are trying to do that with our approach. As I explained here, we're trying to have a biomarker to determine those people. So this is what individualized medicine means. Finding a way, like biomarkers, to tell who's going to best benefit from a specific treatment. And because I have this feeling like all of these drugs may not work for everybody. So we have to, and me and many other researchers are trying to figure out ways to do that.
So correct me if I'm incorrect, but it seems like when I look into the future of treating Alzheimer's and other dementias, that it's likely to become a chronic disease that can be managed or reversed a little bit. Is that kind of the future, like, as opposed to we're going to cure it? Like, what's your take on the future?
I would see it how your first way, that we're going to manage it, and that people could, it would be great if we could get to the point where people can continue living and taking medications that maintain a level of cognition that can help with their lifestyle, right? Because at the end of the day, I think that's the most important. But we've seen I think with other research that's been done to come up with therapeutics for example, for HIV or for cancer, that you can, over time, hopefully not just come up with ways of determining what treatments are best for people, but also, just maintaining a lifestyle despite, having these problems, I think would be amazing.
Great. So we got a question that's the perfect lead-in to this. Is there a way to measure the amount of Kibra in the brain? Now, I know this paper was actually involved human data, right?
Right.
Involved some biomarkers, but talk about the issues. Is there a simple way to know what we've got in our brain?
Right. So from my lab, we have done most of our studies on post-mortem brain tissue from donors, which we, by the way, very much appreciate these kinds of donations for our research. It's so incredibly important. But that does not help with our issue of finding biomarkers. What we're starting to do that's more realistic is looking at Kibra in cerebral spinal fluid from people. So we have detected Kibra in cerebral spinal fluid, and interestingly, we do see that levels of Kibra in the cerebral spinal fluid are associated with cognitive impairment in people with Alzheimer's disease. And also, Kibra levels are highly correlated with levels of tau in the cerebral spinal fluid. So this suggests to us, our recent report in this paper suggests to us that Kibra could be a potential biomarker for synapse function that is dysregulated by tau specifically. We did not see correlations with amyloid beta and Kibra levels, so we think that the effects with Kibra, at least in Alzheimer's disease, are more related to tau pathology than to amyloid beta. So that was kind of the interesting finding we've had so far. We do have an ongoing collaboration with folks at UCSF to work on larger cohorts of people and look at their cerebral spinal fluid and do a bigger study on that. And because CSF is a little bit hard to get, or cerebral spinal fluid is a little bit harder to get, we're also going to start looking at Kibra in blood, because that's much easier for us to use as a biomarker.
And the big project is engineering that therapeutic.
Yes. So this project is something that started, the Forever Healthy Foundation actually started helping us to fund to get it off the ground. So here, since we did a bunch of mechanistic work at the Synapse looking at Kibra and Alzheimer's disease, we discovered that this very small piece of the Kibra protein was sufficient to recover that plasticity at synapses. And the neat thing is that not only does it restore the plasticity of the synapses, but it does that despite having the toxic tau in the brain. And so this suggests that it promotes the functional resilience of the synapses in the context of tauopathy.
So what's it doing for the mice?
Right. So we've treated our mouse models, our mouse tauopathy model, with this KB PEP. This is the little piece of Kibra that we're using as a therapeutic. We've treated the mice. They have what we say is repaired synapse functions that's resilient to the toxic tau. And we're also seeing great memory improvements in the mouse models. So these mouse models, they have memory impairments that are similar to the impairments that humans have. and we can recover the memory with this KB-PEP treatment.
So how do we get that from the mice into people?
Oh, yes. Great question. I would not do that yet. We have more work to do. This is what we call preclinical studies. So ideally, we need to figure out the best mode of administration. The type of drug that this is currently is a peptide, and there are peptide therapeutics being developed, especially for introduction to the brain, which actually, in some cases, we got really excited about the potential of having an intranasal peptide administration, right up the nose, because that could actually give us really good access directly to the brain for this therapeutic. But we still have many tests to do. There's also some like figuring out dosages, and we have not observed adverse effects in the mice caused by the KB-PEP. We haven't seen any obvious kind of bad symptoms. But we do need to continue doing more studies to get at that. But we're hopeful. We're in the process of, my lab is in the process of making a startup company based on this technology. So hopefully that will help with getting that process going.
That's great.
Yeah.
Okay, so let's see. So whoops, I always do this backwards. Hold on. Jeez. Okay, so now it's time for some questions, and I have a bunch of good ones. Some Parkinson's disease, Lewy body dementia aren't listed as tau diseases. How do they differ? Do they?
Sure. I think the different pathologies, there can be some commonalities across these neurodegenerative diseases, but typically it's what type of protein is causing the problems. In Parkinson's disease, there are other proteins that are thought to be the toxic ones. Also, the differences are where in the brain are the problems originating. And in Parkinson's, the area of the brain that's degenerating is different from where in the brain is degenerating in Alzheimer's disease. The first, I think probably if you know somebody who's had Alzheimer's disease or somebody who's had Parkinson's, the really obvious thing is the first symptoms that show up are different, right? Alzheimer's disease, the first symptoms are memory related. That's not the case with Parkinson's disease. So this is indicative of the different mechanisms that cause pathogenesis.
So, as expected, we're getting a lot of questions about lifestyle issues. I'm going to set this up with Tara. So when we did prep for this, I go, so Tara, what should people do? And her first response was give money.
Oh, give money, yeah. That's right. One of the challenges in developing a therapeutic is it's very expensive. So so that was a little bit of a joke. I'm a little sarcastic sometimes.
So I just want to point out though that this woman, she is a serious cell biologist. She is laser focused on getting that drug into the clinic. That said, I'm going to ask you, so people want to know about supplements.
Yes.
Folks want to know about the gut microbiome.
Oh.
Yeah, so there is some lifestyle studies that are happening in mice, but
Yes.
what do they hint at, and what are the issues with those studies?
Thank you. Yes, that's right. There are a lot of supplement studies, like things you can take off the shelf that in the literature that is potentially having effects in mice. Or associated with things in human studies, I think all of this research is important to do. At the end of the day, though, I would say, and this is probably my personal feeling on it, I would be hesitant to really jump the gun on things until we see data from a clinical trial. Clinical trials, when they're done well, are hopefully definitive and can really tell us, is something going to reduce risk for Alzheimer's or not, or is something going to slow cognitive decline or not. And I think the clinical trials are, really the best evidence of whether something is beneficial. So if your supplement you're taking hasn't been tested yet in a clinical trial, then it's like a little bit like, okay, it might or might not help you, potentially. Like, we don't know yet. There's a lot of things that are still a little bit unknown. But science is always advancing. So I still, having said that, I still want to be optimistic.
And how about diet? Because, there's
Yes.
things about high fat, too much sugar. What do we know about, is there anything specifically, even in the mice, that we
Yes.
know about diet and tau?
Right. So there is an association in mouse models between high-fat diet and changes in the brain that would not be beneficial or that could affect cognitive functioning. And so I personally, since I'm not an expert on metabolism, I think it's a little difficult for me to say, that a certain type of diet is better than another type of diet, because I think this is very, this could affect things at the, at that kind of level potentially. But it could be. We need more clinical trials. Yeah.
This is a fun one. How do you give a mouse a memory test?
Oh, yes. It's. I think most people would be surprised to know how well some mice can remember things. Well, or maybe not, because you have mice in your house and they're sneaky. But we have a number of behavior tests that we do on mice. One of them in particular is called the Barnes maze, where mice are put on kind of this round table that has a bunch of holes, little holes for them to crawl through. And only one of those holes on the round table has a little escape box. And the mouse, the test is that the mouse has to use what we call spatial cues on the wall. So we put little posters of like a square and a star and a letter A, something like this on the wall. So the mouse can see it and can learn where the location of that escape hole is. And so we train the mice every day. Maybe they get 2 or 3 trials a day where they're put on this platform, and they are supposed to find the hole. You can use a reward system, meaning like put a little treat for them in the box for them to learn how to get in there, or use kind of a verse stimulus, which would just be like a noise in the room. And mice, if with a big noise, they want to go run away and hide in a hole. So these are kind of motivations that we use. And over time, they learn very quickly where that hole is. And then at different days, after learning where the hole is at different days, you can then assess memory. So how long do they remember where that hole is? And there's certain ways we can quantify that. So that's an example of one.
Another question, good science-based question. With Kibra, how are you guys looking at overcoming the blood-brain barrier? Because that's always been an issue in neuroscience.
Yes.
It's good that we have that barrier, but it can be, you have a hard time getting things through it.
Yes, that's a great point. It's something we think about a lot. So the Kibra therapeutic we have, it's a pretty small peptide. Right now, it's 20 amino acids. Amino acids are the building blocks of proteins. So we think it could be small enough to get through the blood-brain barrier. Other peptides that people have used have shown effects that, through systemic injection to getting into the brain, so that's encouraging. We are just starting to test this. So yeah, ask me again in 6 months.
Okay. I also got questions about people who are interested in the possibility of doing brain training, brain games to address dementia, perhaps address tauopathy. What does the research hint at?
Yes. I think there's our association studies showing that training, maybe I'll just say training your brain, or giving mice extra training can ameliorate some of the effects that are associated with cognitive impairment. I think again that these are kind of association studies, so we interpret them in that way, and that whether this is going to treat dementia, I think could only be determined through a clinical trial. But I also, when I think about the brain games, some of them are online. I'm a parent. I don't like my kids being online a lot, so I would say, I would not recommend playing video games for hours, thinking that that's going to treat dementia. But talk to your doctor.
That's great. So we've got questions about those GLP-1 drugs, all the weight loss drugs that are showing promise, perhaps, and it kind of a corollary. Whatever you can say about that, but do you think that there are existing drugs out there that are already FDA approved, that are being used for other things that could be helpful for treating dementia?
What I can tell from the field, like those of us studying Alzheimer's, I think that there's not much, not much signs right now that these are going to be, therapies that are necessarily going to make really big effects. Maybe, so here's the thing, potential thing to consider. Maybe some things modify risk. So when somebody says you might be at risk for Alzheimer's disease, we do know things that make people at risk. For example, genetics, right? The ApoE4 gene is definitely associated with risk for Alzheimer's disease, for example. But this does not necessarily mean that you will get it. So it's potential maybe that some of these things that you mentioned, Chris, maybe they could affect risk, but that doesn't necessarily mean that you would still get it. It would just reduce your risk a little bit, something like that.
Great. Well, I want to thank you for being here today and talking to us. This is really exciting stuff, folks. I got to tell you, when you talk about making an end run. Oh, I know, one final thing that we talked about. So you're looking at this Kebra protein in the context of Alzheimer's, but you're also here at the Buck Institute for Aging.
Yes.
How are you including aging in your work?
Yes. So we are looking at Kebra in the context of aging as well. As I mentioned, that genetic polymorphism is associated with just memory decline in aging in general. And so we do have 2 projects in the lab working on this. We have tested the Kebra peptide in old mice, and we have seen beneficial effects in that context as well. So there are a number of other studies we need to do to determine whether it's through a similar mechanism as in the tauopathy model. But it is potentially really exciting if this approach could be applicable, across conditions to some degree. So yes, we are working on normal cognitive aging as well.
Great. All right. Well, thanks to you, Tara. Thanks to you folks for coming. Here's what's going to happen next. So as I mentioned earlier, you're going to get a follow-up email with links to some background material and that video about concussions, which I really do encourage you to show to any young person who is considering contact sports. I mean, seriously. Okay, so I'm going to be back here on January 15th after the new year. I will be talking to our president and CEO, Eric Verdin. He'll be talking about the science of longevity and kind of fact versus fiction. I saw him in the back of the room. I thought he snuck out. Maybe you guys could get your questions in early, but I think you're going to have to wait. You're going to have to wait for January with that. We got a question from somebody about, could people in Marin get involved in our research? The answer to that question is a huge yes. So we have a new clinical research program here at the Buck. We've done really great jobs in making worms and flies and mice live better longer. Now we're moving that research to people. So we have several clinical trials. We are recruiting. If you'd like more information, you can find it online, but outside in the atrium, after we're done here, there are going to be folks from our clinical research team who are available to answer your questions. So please take advantage of that. I would be remiss if I didn't make the fundraising pitch at the end of these events. As Tara mentioned, research is expensive. One of the advantages of giving here at the Buck is that your research does have an impact. This is a photo of our very popular donor group called the Impact Circle. They have also supported Tara's work in trying to get Kebra out of the lab and into people. So it is that time of the year when folks are thinking about charitable contributions and taxes. So just want to let you know there's a way that you can save on your taxes, especially if you have a 401k. There are ways you can save on your taxes and support the Buck. So folks from our philanthropy department are going to be out in the atrium afterwards. If you have any questions, no commitment, but just ask them questions if you have any questions about how this works. So going forward, I'm assuming that intellectual stimulation is good for the brain. You can learn more about aging. We have a very popular online learning class. It's getting great reviews. You can find it on our website. Please check it out. We also have a podcast called We're Not Getting Any Younger Yet that focuses on interviews with leading researchers from around the country. So I encourage you to check that out too. If you're new to the Buck, or even if you've been on a tour before, please sign up for one of our tours. They happen on Tuesday and Thursday mornings. We have a new museum exhibit, so you can in addition to enjoying this gorgeous IMPA design building, you can also learn about the trajectory of research on aging and get the backstory of the history of the buck. So you can keep up with the buck. Follow us on social media, sign up for our newsletters, and just keep in touch. So once again, I want to thank you for coming. I will be here on January 15th. I encourage you to have the most wonderful, healthy holiday season. I'll see you on January 15th. Come with questions for Eric Verdon, both about what's happening at the Buck and also in the larger realm of research on aging. He'll be the guy to answer those questions. Thanks for coming. Happy holidays.